السلام عليكم ورحمة الله وبركاته. معكم دكتورة زينة مكي عبد الكريم، تدريسية فرع الأحياء المجهرية. راح أدرسكم هذه السنة مختبرات الجراثيم العملية. Our lab today concentrate on culture media and sterilization. The main objective is to know what is culture media, what are the main ingredients of culture media, and what are the types of culture media. Culture media is the nutrient substance that's used for isolation, identification, and growing of microorganisms. So it is a nutrient substance for microbes. And we have different ingredients for culture media according to the metabolic pathways, the requirement of the microorganisms. The main ingredients are peptone water, sodium chloride as a source of minerals, nitrogen source like ammonia, carbon source like carbohydrate, protein source like beef extract, and what is called agar-agar. Agar-agar is a golden yellow granular powder. It is used for preparing solid culture media. So it is a solidifying culture media. Its melting point 92 to 95 degrees centigrade and has a solidifying point which is 42 to 45 degrees centigrade and has a concentration of 1.5 to 2% for preparing solid media and 0.4 to 0.8 for preparing semi-solid media. We have different kinds of culture media. According to the physical activity, either we have a solid culture media or we have a liquid culture media, like this. This is a nutrient broth, an example for the liquid culture media. And these all are examples of solid culture media. Starting with the nutrient agar, it is the most simplest one, and it is used for cultivation of most of microorganisms. And its main ingredients are peptone, water, beef extract, and sodium chloride, together with the agar-agar media, agar-agar substance. This is the nutrient. We have other examples, is the blood agar, for the solid media, chocolate, this is the chocolate, has a brown color, this is the chocolate, while the blood red in color. The chocolate is a heated blood, while this is the blood only, without heating. These are examples of solid culture media. This is mannitol salt agar, and this is makonki agar. And this is the mannitol salt agar. الأورنج كنا شوية على أورنج هو المانيتول سولت agar، شوية على بينك هو الماكونكي agar. Culture media can be put in a petri dish or in a glass tube. We have different kind of culture media according to the uses, as simple culture media, nutrient agar, or enriched culture media as a chocolate and blood agar. It is called enriched media because it's a simple media added to it enriched substance that enhance the growth of fastidious microorganism, البكتيريا العنيدة. مثال على المواد اللي تعمل enrichment للculture media: blood, sugar, acidic fluid, or serum. So example on enriched media is the chocolate and the blood agar, because they contain the blood that enhance the growth of fastidious microorganisms like genus Streptococci. Another type of culture media is the selective culture media, like mannitol salt agar and makonki agar. The selective culture media means it contains substance that enhance the growth of certain microorganisms and inhibit the growth of other. So mannitol salt agar is selective for the Staphylococcus aureus, and other species of genus Staphylococci cannot grow on this media. And this is the makonki. It is also a selective for the gram-negative bacteria because it contains bile salt that inhibit the growth of the gram-positive bacteria. Makonki agar also is a differential. So we have another type, differential culture media, like the makonki. Makonki can differentiate species of a gram-negative according to the effect of the gram-negative on the culture media. So differential means it contains certain species produce certain effect on the culture media. So gram-negative bacteria could be lactose fermenter, produce pink colonies on the culture media, on the makonki, so they are differential for the gram-negative for the lactose fermenter, while others, non-lactose fermenter, appear as yellow colony on the McConkey. So McConkey is a selective and differential culture media. Another type of culture media is the Muller-Hinton agar. This Muller-Hinton agar is used for antibiotic sensitivity test. The method is we take the Muller-Hinton agar inoculated with the microorganism that we need to test it for its sensitivity for the antibiotic, and we put the antibiotic disc. These are antibiotic disc. ذول الانتيبيوتيك ديسك يكونون جاهزين وبكونسنتريشن معلوم حسب الكتل اللي متوفر بالمصانع اللي صنعوها. After 24 hours of incubation, we see that some of the discs are surrounded with a clear zone, others are not. This disc, for example, surrounded with a clear zone. This disc, smaller one. This disc is smaller. This is without any clear zone around it. So, which means that if there is a clear zone, means that the microorganism is sensitive to that antibiotic. But if there is no clear zone around the antibiotic disc, so the microorganism resistance to the antibiotic. According to the diameter of the clear zone around the antibiotic disc, we can know how much the microorganism is sensitive or resistant to that antibiotic. This is our second part in the lab about sterilization. We have in sterilization applying heat, either dry heat or moist heat. This is an example on the dry heat. This is the benzene burner, or it's called alcohol burner. We can by it sterilize surgical instrument or some bacteriological instrument like this bacteriological loop. We put it at 45 degrees until it become red in color. So benzene burner or alcohol burner is an example on dry heat as a method for sterilization, one of the physical methods for sterilization. Second method is applying moist heat. An example on moist heat is the autoclave. هذا الجهاز يوفر لنا method for sterilization عن طريق steaming و pressure. So apply pressure with steam to kill the microorganism, especially the spores of the microorganism. It has a temperature up to 121 and it has a pressure up to 15 IBING, and it has a time for about 15 minutes. We can by it sterilize surgical instrument, bacteriological instrument like loops or media or culture media, or even some dental instrument. And this is the best method for killing the spores.